Hi, would you like me to start now?
Yes, sir.
Okay, great. Thank you very much for the invitation. I guess I have the student community of the Science Club to thank for this. And it's an exciting topic that they have chosen today on Science Day. But actually, in the Science Week or month, I think is what they have planned to talk about symmetries. And what you're looking at here is an image which some of you may be familiar with. Um, It's a woodcut by an artist called M.C. Escher, who is renowned for having made tiling patterns very popular in the West. Uh, in fact, in this particular tiling pattern that you observe, he has also tried to depict depth of perspective, where as you go from the center outwards, there is a reduction in the size of those structures, but in a manner that appears to scale with the size. And the two circular dots, in fact, are supposed to represent something like eye spots of an organism called Planaria. And again, some of you who are reading biology might have heard about it. Suffice to say, it has fascinated biologists for almost 100 years, because Planaria, when it is cut into about 1,000 pieces, generates a new organism from each of those 1,000 pieces. So in that sense, self-similarity, tiling, patterns, and symmetry, they are intrinsic to the physical nature of biological systems, to what we observe around ourselves. And that is why my topic today is indeed symmetries in biology, from organisms to molecules. And I hope I will be able to do some justice to this, although there are many themes I wanted to touch upon, but I've decided to focus a little bit. Um, but the focus is, in fact, going from symmetries to lack of symmetries. And what you're looking at here is a monograph of, I'm sorry, a picture from a series of images created by Ernst Haeckel in his monumental work called Art Forms of Nature. It was published in 1904. And what you're looking at particularly here are organisms of the phylum Cnidaria, class Anthozoa. Specifically, these are sea anemones, undersea organisms, usually marine. And I hope you notice that almost all of them along their long axis, or you may say perpendicular to their long axis, they have a radial symmetry. It's obvious. Some of these radial symmetries are restricted to a few degrees, some to many degrees, some have minor asymmetries in them. Uh, does it have any meaning? Does it have any connection with um, how they got here? And can we tell something about it? That is something I'm going to try and dwell upon in the coming few minutes. In fact, if you go to a related Cnidarian, Scyphozoa, which are the so-called true jellyfishes, you see a much more specific manifestation of these radial symmetries. Something that I don't think anyone needs too much arithmetic to figure out what is going on, right? For instance, in some of these jellyfish, you can claim that there are something like, depending on the number of arms, that many, that by one by two, the number of rotational symmetries possible. But as we go up the evolutionary tree, and by up or down, I don't mean anything other than just simply to more recent organisms, like vertebrates. And in this particular case, in the vertebrate phylum, if we go to amphibians, so this is phylum Chordata, class Amphibia, there is something of a similarity to be seen in features between different frog species. And what you observe here is, of course, the webbed feet, the snout, the tadpoles are depicted, and a couple of different variations in color and shape and stripes, but broadly similar structures. What As far as symmetry is striking, of course, that they are bilateral symmetry in external view.
Sir,
And this has led, yes,
I'm sorry to interrupt, but I think the slides are not changing.
okay, I have no idea why. um, oh, I think I know what happens. This is the usual problem with Google Meet. Okay, let me try again. Do you now see my slides changing?
Yes,
Organizers, do you see my
yes, sir.
slides changing now? Okay, thank you very much. Thank you for letting me know. So indeed, I was talking about the presence of radial symmetries in a diverse set of organisms, which when we go to more complex organisms, vertebrates in particular, we seem to find to be missing. Indeed, they appear to get restricted in their degrees of symmetry or the axes over which we can find symmetric structures, and bilateral symmetry essentially implies we have only one axis of symmetry. Uh, Many experimental studies that have tried to investigate the how, meaning the molecular basis, the cellular basis, the developmental basis of these symmetries, have used amphibians and other vertebrates as model organisms simply because we can't make experiments of the kind that we can do on smaller animals than we can do on humans. This is ethics issue. Um, But funnily enough, this is not enough to ignore the fact that the plant kingdom, so as opposed to the animal kingdom, also shows dramatic symmetries. And you just need to take a walk in your garden or look at your potted plants to remind yourself that you can find spirals and whorled patterns which have been broadly classified into these descriptive phyllotaxy patterns emerging spontaneously and heritable, meaning to say that the arrangement of leaves, whether it is one at a time on a stem in a certain sequence, or many multiple, two or more leaves at a node in a specific manner, are conserved for a given plant species. Indeed, a lot of work has gone into trying to understand what is the basis of these phyllotaxies and whether there are even mathematical models that can help us understand the mechanism that gives rise to this. So for the rest of my talk, I'm going to try and dive a little bit beyond this, what is possibly familiar to you from high school biology, descriptive biology, to something a little bit more about the potential underlying causes. And in that sense, maybe my slides are already playing up a bit, sorry. Mhm. The amphibians that I spoke about earlier have provided some beautiful examples for how we can think about how these dramatically symmetric structures emerge. So I'm going to play you a video now, which is that of a developing embryo, and it has some voiceover in it. So I hope you will hear some of it also.
This video shows the remarkable synchrony of the early embryonic cell divisions for the clawed frog Xenopus laevis. About 25 frog eggs were fertilized simultaneously in this petri dish, and then filmed over the first dozen rounds of cell division. Each cell division cycle takes about 25 minutes in real time. Not only do all the cells in each embryo divide simultaneously, but the entire dish of embryos maintains essentially the same clock time over several hours after fertilization.
Right. So what I was hoping you would have noticed, even beyond the synchronous nature of the cell divisions, is the fact that the divisions themselves are regular and result in patterns which take us from what was initially a spherical structure, a spherical object that you see here, to a later stage which gets reduced in the degrees of symmetry that you can describe it by. In other words, you can say the degrees of freedom get reduced in terms of the organization. So I'm going to make my first sort of postulate, and this is now partly because of a lack of work, maybe concretely on these kind of topics, to say that as far as embryological development of higher vertebrates goes, it would appear that embryogenic development of multicellular organisms, particularly vertebrates, typically seem to reduce the degrees of freedom or go from radial or high degrees of symmetry to very low degrees of symmetry where we have only just one axis of symmetry for that matter. And we'll see if this is true, and we'll see what else also comes out of it. Now, what you're looking at here is a very unusual system to talk about in a biology talk, but suffice to say, every one of you has had an opportunity to play with soap bubbles. These soap bubbles that you're looking at here, it's from a paper published in Science, are the creation of a computer. They are a numerical simulation using what is called multi-scale physics. The idea is to employ mathematical calculations across space to simulate multiple processes, in this particular case, membrane tension, fluid drainage, rearrangement of these cell-like structures, the bubbles, and merging and splitting. Yeah? And with that, they hoped to understand how a foam or a collection of bubbles behaves in time. Their purpose was, of course, to try and develop methods, numerical methods, that overcome some of the challenges that have, in a way, slowed down progress in understanding foamy patterns, foamy structures that are out of equilibrium. But for me, the most exciting part about this is that the out of equilibrium nature of soap bubble forms is exactly what we see in biological systems. So I'm now going to play this video and frame a few questions on the basis of it. Yeah? So the time is in calculation time. It took much longer in real time. What you should start seeing is that these soap bubbles at some point start merging, bursting. There's a loss of fluid. And somewhere, you start seeing them going from this very complex set of bubbles to only a few that appear to be more and more regularly arranged in very simple lattices. You could say almost simple symmetries, fewer and fewer. Fewer and fewer cell bubbles. And these bubbles, in that sense, almost appear akin, similar or comparable to cells themselves. In fact, this process of going from a very complex foam of bubbles to a few very simple, almost spherical bubbles is exactly the reverse of what is happening during embryogenesis, as you just saw. Right? We had started with a single spherical cell, and we went to a complex, I'm going to say broadly, foam-like structure. And If it took this long to get the numerical computation of just non-living material, yeah, to make it predictable to match with experiments, then you can imagine that the cell, which is not just an air bubble, but has complex genetic programs in it and machinery and biochemistry, must be much, much harder. And so we're going to sort of dwell a bit on how to make the problem a bit simpler. And as you see in this form, our symmetries are now restricted to probably, we can say at this point we have three axes of symmetry probably in 3 dimensions, and we'll very soon be down to radial symmetry. There we go. Okay. Now, I'm going to call this life in reverse and try to give you some evidence for why this is so. Yeah. So embryonic growth, at least in more primitive organisms, follows the kind of division patterns in the first 2 divisions, from 1 to 2 and 2 to 4, of orthogonal planes, 90-degree planes of division. Whereas in mammalian embryogenesis, these planes are not the same. There's not just a 90-degree switch, there's also positional change in the planes of division. Suffice to say, this already means that you are getting very soon to reduced degrees of rotational symmetry. In fact, if you open a developmental textbook, developmental biology textbook, and this image is taken from the standard book in the field called Developmental Biology by Gilbert, you see that there is a whole range of patterns that you can observe going from one cell to a many-celled organism at the stage of embryogenesis. And this led people like Ernst Haeckel in the early 1900s to postulate that ontogeny recapitulates phylogeny. In other words, developmental patterns, ontogeny, yeah, are in some senses comparable to the process of evolution itself, evolutionary diversification. And this is, while this is partly true in a qualitative sense, in a quantitative sense, things get more complex because the nature of embryogenesis and embryonic developments, as far as these structures are concerned, already in incomplete cleavage, the green boxes, begin to look far less like anything in the previous so-called more simple organisms that you see in the yellowish and orangish boxes, right? And so in a way, if you want to make sense of this, we have to go back to something simple, at least to try and ask some questions about how do these structures form. So what we are looking at here is a sea urchin adult at the bottom of the ocean. It's a very beautiful organism, and it has degrees of radial symmetry, which you can qualitatively observe from above. But if you look more carefully at the embryogenesis, you will find that they indeed go from that first spherical egg. So everyone was one egg at one point in time, to a two-celled stage, B, to a nice almost cloverleaf structure, C, and then an additional plane added above it, and things get fairly complex from then on, and almost you start seeing in 3D a loss in the degrees of symmetric axes that you can find, to a point that in the K, which is the late embryonic stages, you can only find a few degrees of symmetry around axes that are perpendicular to the vertical axis in this particular case, right? So the body axis symmetries appear to be either bilateral, radial, or not at all in most animal systems. But what about us? What about human beings, right? We argue that we are bilaterally symmetric, and this is true. from the external perspective at least. And this artist's view depicts to us the picture that is famous from the drawings of Leonardo da Vinci, a medieval European Renaissance artist, um, demonstrating the measures of the human body. And the thing that I, of course, want to emphasize is that the attempt at demonstrating bilateral symmetry. And yet, even in his drawing, the structure is not exactly bilaterally symmetric. Not to mention the fact that he has depicted one leg pointing one way and the other, and the sexual organs are a bit deviated, and the face is not straight enough, and many other small details. Besides these small defects, there's something that is more than just that goes beyond the superficial or skin part of it, which is the internal organs. So if we dive under the skin of a human, we know very well that we are left-right asymmetric. This so-called normal arrangement in the Latin term for medical phenomena is called situs solitus. Yeah. Now, the question, of course, arises that if we grow from that one egg cell and everything is symmetric, and at least we have bilateral symmetry in the embryonic development, then why do we get suddenly one organ only on one side, not on the other? It's unfair, right? Why does the right wing have only the heart, or the left wing only have the heart, sorry, and only the right wing have the liver? Um, so people have been asking this question for a very long time, and it was only in the recent years, due to dramatic technical improvements in microscopy, in molecular biology, in our ability to understand things and bring all these concepts together, that it appears that the process by which this asymmetry in the human organ placement is generated has something to do with okay, let's see if I can kill some programs, prevent this from going away. Guess I can't stop the Google. Oops. That's all I can stop. Okay. Perhaps this will still work. So it turns out that in a few rare cases, there are people whose heart is to the right and they have breathing difficulty, and at the same time have a high prevalence of male sterility. Uh, this is, in fact, has a name. It is called Cartagena's syndrome, after the person who discovered it. And it suggests that organ placement is not always as we consider to be normal, right? So the clue came to the answer of how this happens from genetics, because mutations were found in the humans, and in order to ask the question whether this one mutation is sufficient to reproduce this effect, experiments were performed using mice, where we can do these kind of directed genetic experiments to knock out the two copies in a diploid organism, that is to say two chromosome copies therefore diploid, the gene for a protein called KIF1b. I'm sorry, KIF3b. It should read KIF3b. KIF3b encodes for a protein that produces molecular motors. And what they observed, what the people who did this work, Hirokawa and Nonaka, was that at this stage of development of the mouse, at comparable stages, the tail had not yet turned. And even more interestingly, the heart involutions, which you see in the bottom panel, had not gone in the correct direction. And this was kind of a clue.
I can't see your slide, sir.
Let's see. It's a little strange.
Oh, yeah, it's visible now. It's visible now.
Oh. Do you still see them?
Yes, sir.
Are they changing?
No, sir.
Okay. So, as I was saying, in the Cartagena syndrome, there is a left-right swap, and this can be reproduced in a mouse model. In other words, doing experiments of genetics, of knocking out a gene from a mouse, you can reproduce an effect, which also led the researchers to a finding about what is the basis of such a swap in organs. And ironically, it has nothing to do with the organ itself. It turns out that there is a structure called the node, which is magnified here from the 7.5 day embryo. And this leads, when you look at it in a higher resolution at 20 micron scale, the middle panel here, shows cilia present, these white structures, which are highlighted in the upper right panel image. What the researchers had found was that these cilia in the knockout mutant were absent altogether. Now you might ask, what do these cilia have anything to do with anything? And cilia are related to something called a ciliary motor. This is the basis of how your airways are kept clear, how your digestive system works, how your retina function. And it has something to do with a motor called KIP3A and KIP3B. These are the light blue, dark blue proteins that you see. And the fact that they produce transport inside the cilium.
Sir, sorry
Yes?
to interrupt, but the
Yes.
slides are not changing.
Okay.
Yeah, it's visible now, but they are not changing.
Okay. Let's see. Do you see my screen?
No, sir.
It will take a few seconds.
I know that. Yeah, so just wait a minute. Do you still see my screen?
Yes, sir.
Yes, sir.
Do you see my slides changing? I'm changing my slides now.
Yes, sir.
Yes, sir.
Okay. Okay. Thank you. So I don't know where you last saw this, but at 7.5 day embryos, we find that the ciliary motor is the one that seems to be defective. And it turns out that the cilium, which produces a sort of asymmetric beat, that is to say what is called the beating stroke or power stroke and recovery stroke are different. The return is different from the, it's like a overarm swimmer, so-called crawl in Olympic swimming. The upstroke is a long arching stroke, and the backstroke, the pull back, the recovery, is a short one. The arm is short. And this has to do with the nature of the structure of the cilium. And these nodal flows appear to create a direction of flow of molecules that themselves are ligands for a receptor. This is what is marked in R in this diagram. That results in signaling and produces an organ at the right place. Yeah. So your left, your heart in your left side, and liver on the other side. And in the absence of the cilium, as in the lower panel, essentially it results in a randomization, which is why you get 50-50. Some people have the left, some people have the right. Now you must of course ask, why is it at all decided? Why is it not in the middle somewhere? Well, for that it turns out there are signaling pathways downstream that force a decision to go one way or the other. The choice of the decision is driven by these ciliary flows. So in some senses, these structures at a molecular scale, nanometer scale, are deciding our meter scale or a few centimeter scale organ positions. And this is fascinating. So this for me is a truly multi-scale system where we go from a few nanometers of molecules which seem to generate small asymmetries that develop in result in the formation of these higher level asymmetries. And what you're looking at here is an experiment that demonstrates the formation of spontaneous spirals with exactly the same molecules, kinesin, which I mentioned earlier, and microtubules. And these are responsible also for the formation of your spindle. What you're looking at here is the spontaneous transition from a radial to bilateral symmetry by the action of microtubules and motors to form these bipolar structures, which are very familiar from the textbook picture of spindles. And these, in the case where there is no where there's no cue for the bipolarity, can spontaneously generate these structures. And this is some work we had done earlier. In the interest of time, I'm going to speed up a little bit. And indeed, this kind of spontaneous structure formation is so reproducible in experiment that we went ahead and did some more numerical computational calculations, sort of like the ones that had been done in the case of the soap bubble foams, except that these are not multi-scale. They are all at the nanometer scale, and they give rise to collective behavior at a micrometer scale. And what we observed was that in this movie, you see these radial structures of microtubules interacting with kinesins or dyneins, being either, so minus-ended motors or plus-ended motors, being either forced to form what is in the middle, regular lattices of nucleation centers, or a central radially symmetric aster. Indeed, we found that when we now take all these components and put them together in a simulation, we can generate a whole range of patterns with no pre-patterned template, you see. And so this kind of spontaneous onset of patterning and rotation and movement for us is a very surprising feature of structures that inherently have no symmetry in them as such, but give rise potentially to either symmetric structures or break the symmetry of the structure. So in summary, I'd like to emphasize that while biological organisms themselves at a microscopic level are symmetric, we have reason to believe that there's an underlying asymmetry that drives the processes and makes them transition from high degrees of symmetry to lower and lower degrees of symmetry. And the more recent the organism, the more complex its cellular nature, meaning diverse tissues and so on, the more it seems that these symmetries seem to be reduced. Many of the things that I've talked to you about are collective effects. They are not down to just one molecule, but due to the ensemble outcome of what happens between these molecules. And it is a combination of mechanics, biochemistry, and genetics that brings these processes together, and they're spontaneous. In fact, it turns out randomness is a very important part, surprisingly, in the determinism of patterns. By determinism, I mean you always get, with the exceptions of people who don't have the good fortune of having them, you always end up with 5 fingers on each hand and 2 hands or 3. And indeed, many measures of symmetry have been brought out, which help us understand this. But in some senses, in a mathematical sense, we are still far away from making sense of what goes on in biology because it's a little more complex than that. So I'm going to stop here, and just to say that the work I talked about from our lab is mostly done with a PhD student and now postdoc of mine, Neha Chaitanya. Okay. And I'm happy to take any questions if there are any. Thank you.
Thank you, sir, for this talk. It's an honor to have you with us. I would like to mention one of the recent finding of sir. It is the use of a computational tool for a single molecule observation via some CTC microscope. So it's one of the most interesting thing I have come across. Like I read that article yesterday only. So he's a biophysicist and he's working in the field of microtubule motor interaction and self-organization in cellular patterns. So yeah, thank you very much, sir, for this talk.
Thank you very much. So I think if there are no questions, I'm going to log off, and I wish you the best for the rest of your Science Day extension. Right?
Thank you, everyone.
Thank you. Bye-bye.
Thank you, sir.